let's talk about the brain, always a good idea. In more particular terms, whether and how brain science, as we think, could still help us, maybe, with arriving at better machine learning, at better artificial intelligence. Let's see. So when we look at images like this, we are pretty much aware that we are using neurons, nerve cells, to process this image, detect that it's a face, maybe even, memorize or remember that this may be a composer, and depending on your interests, maybe even identify the name of that composer. Now, the same is very well possible with a noisy image, no problems. And as you all know, today's machine learning trying to solve these problems is also using neurons in some way or other in what we call a neural net. But obviously neurons implemented in silicon and pretty inefficiently implemented usually. And then network architectures like this, as you all know, are still at the basis of what is called today's deep learning, at least a large fraction of it, and definitely when it comes to image analysis. Now, why is that called neural, and what is brain science to do with it? Well, as you know, there are two things that came from biology as inspirations. One, nonlinearities of the neurons, later understood to be very important to be able to approximate almost any function. And then secondly, and this is often overlooked, the feedforward connectivity. This came from, as an inspiration from the visual system mostly, the idea or the analysis in the first anatomy, if you want, of the last century, that retina projects to then the later visual centers via the thalamus to cortex, and then it's delivered further and further into cortex in the human and other mammalian brains. Now, this is, of course, true, but it's not the whole picture, and this is what I want to talk about today. Now, artificial intelligence today, at least to a very large fraction, as I just said, is deep learning, meaning many layers, meaning not so many layers of a feed-forward perceptron. Nothing really fundamentally different compared to what was known and proposed in the 60s of the last century. And definitely you could say whatever we're doing right now in artificial intelligence corresponds to old neuroscience in the sense that I just explained. Now, obviously, I don't want to ignore, there are more things to it. Learning rules, backprop, as you know, has some claim, but I don't think any too plausible biological complement. My view is I don't think backprop has a natural correspondence. Others see it differently. And then obviously there are attempts to go to more recurrent networks, and you all know famous LCM learning rule that allows us to deal with RNNs at all. It's the one learning rule that we know of. Again, very difficult to see biological parallels there. Now, why am I talking about this? You could just say, come on, you don't have to build a brain to build intelligent artificial systems, right? After all, we don't have to build birds to fly. And that's an important thing here to see. I'm not claiming that it's necessary to look at brains to improve artificial intelligence. But what I am saying is maybe it is worth looking at it again, and I'll tell you a little bit about what I mean. Again, so luckily our planes are not as flappy as a bird's wings, but anyway, mostly at least these days. Now, what are the key differences between biological and artificial intelligence today? The first one is very obvious, everybody talks about it, energy efficiency. And actually we haven't been talking about it so much because currently we can still fuel the compute centers that are needed for current AI. But it's actually truly limiting if you think about much more powerful computations. It's enormous amounts of energy, and it's nothing compared to the beautiful sandwiches you just had maybe already downstairs. Right? I mean, the energy consumption is just it's orders of magnitude different. But I think there's another difference beyond learning rules and so on that is even more important, and that's also not so often talked about, at least I think is very important. This is label efficiency. So when we learn today's artificial intelligence, we're essentially abusing the fact that we have too many labels, and we're really not making use of labels very well. What do I mean? Well, if you think about it, how does today's AI system learn to call a tree a tree? What we do is we present images of trees, not just one, present another one, and another one, and then maybe some with more than one tree, and then we do that over and over again. And usually actually with exactly the same images, importantly always with a label attached to it. And the worst of it is this, billions of times, as you all know. I mean, when you train a CNN, that's what you do. Maybe a little bit of variation added to it, but you're presenting this poor CNN with this kind of input. And all of you know that is not how we learn. If you've ever thought about how you learn to call a tree, I think my estimate is you heard the word tree probably 50 to 100 times before you were able to tell a tree from a car, let's say, at age 1.5 years. The reason being, you've seen millions of trees, but you've used your labels much more efficiently. And label efficiency is really, I think, the biggest problem we have, or inefficiency in today's AI, and this is something where the brain's much better. Now, I'm a neuroscientist, so what I should be telling you about now, and I will do this now, is what we do in neuroscience, and why I think what we do could help, you hear the conjunctives, could help understanding better what's going on in brains and why, in the end, learning is as efficient as it is, as one of the aspects. Now, what we do is called connectomics. So we care about the networks in the brain, and we measure them. Because they are beautiful, they're also difficult to measure. We have nerve cells, these beautiful structures, if you go at high resolution into your brain. This is just one of them, with a cell body, and in white you see the dendrites, as it's called, the input side of the nerve cell. And then in a second you will see in blue the axon, the output cable of just one nerve cell. It's not even all of it, it's just the local fraction of it. All this branching, one nerve cell. Okay, so this is really what we're dealing with, and if you want to know how many of these you have, the answer is roughly 86 billion. Okay, so why are these structures so beautifully complicated? The reason is that each nerve cell, and this is shown in the upper left corner, each nerve cell in our brains connects to about 1,000 other nerve cells. And if people ask me what's the most important number about brains, it's not so much the number of nerve cells, I think. It is the connectivity of single nerve cells. So the main number about brains is that we have an order of a connectivity degree on the order of 1,000. Each cortical nerve cell connects synaptically and specifically to about 1,000 other nerve cells. This is not paralleled, as far as we know, by any other system today, especially because it's long-range connectivity mostly. It's not local. When we talk about the nearest neighbor of a nerve cell in terms of communication, this nearest neighbor can be at the other side of the brain. And that is really a very, very specific setup of a complexity that we haven't embraced at all, I would say. And we're trying to do that by measuring this. We don't know how these networks are structured, at least not in high resolution and high detail. Now, why are nerve cells so difficult to map? The reason is they are morphologically very difficult. They are very thin. The cables I talked about, the axons, are easily 50 or 100 nanometers in diameter. That is 1,000 times thinner than our hairs. And the problem is we don't have just one of these cables. We have, again, the cables of 86 billion nerve cells in this piece of space that we call our brain. Pretty limited. I mean, it can't grow much more. And the result of this I will show in a second. Now, we are using techniques involving electron microscopes to analyze these structures. Again, we essentially take a biopsy of brain tissue, embed a small piece of brain in plastic, stain it with heavy metals and the like. And then we do experiments like this, where we use scanning electron microscopes to image the surface of tissue blocks, then have a diamond knife that scrapes off the tissue we've just imaged, and then take another image and do that over thousands, tens of thousands, hundreds of thousands, ideally, sections, so we get to substantial distances in 3 dimensions. Because nerve cells are not in a plane, like in the image, they're in 3D, and 3D high-resolution imaging is seriously difficult. Since about 15 years, based on techniques, I should say, developed in Germany, in the Max Planck Society, not in the US, not in China, we are able to do this. And here are examples of such tissue data. So this is a piece of retina, actually, so the first visual processing step in mammalian nervous systems, and which contains already very interesting filter computations of the visual signal. Roughly 30 parallel filters of the visual world are transmitted to our brains, so it's not a single pixelated version, it's roughly 30 as far as we know today that is sent from the retina, and this is computed in the piece of retina I just showed. And this was our first target, and this was a study where we, for the first time, were then able to get a mammalian connectome, as we call it, a small piece of nerve tissue from the mammalian brain analyzed. Here there are cell bodies, so the somata, the cell bodies of nerve cells involved in the visual system, photoreceptors, and other types of nerve cells. And then we reconstructed them densely, meaning all of the about 1,000 nerve cells in this tissue block, and all of their arborizations, and all of their synaptic contact points. Here you can already appreciate what it means to talk about dense tissue, highly packed
extremely densely packed nerve tissue and the cables within. Here's one of those nerve cells, still intelligible, still visible. But then if you superimpose all of about, again, 1,000 nerve cells from this piece of tissue, then you're starting to see what the problem is. So this is the density of circuitry used in a small piece of, in this case, mouse retina to analyze the visual world just at the first stage. So this is, again, the local and sometimes not so local processing used to compute these about 30 to 40 parallel filter representations of the visual scene that are sent to thalamus and then higher order visual centers in the brain of a mammal. And more people realize that the same or roughly the same may be actually happening in higher mammals like primates and us, which was for a few decades, I think, lost, but I think this is coming back, that the same kind of processing is happening in our retina. Okay, so this was a feat, and we were then able to compute the connectivity matrices. This is already a little bit abstracted here, not at a single cell resolution, but using cell types. It was a major step for us, at least, because now we were looking at communication maps of one tiny, but at least finally mammalian circuit. What are we seeing here? This is an adjacency matrix, just a neighborhood graph represented in terms of neighborhood entries in a neighborhood matrix, and essentially indicates who talks to who. Very much like we could create a connectivity matrix of this room here. Currently nobody's talking, but if you were, we would easily see who's friends and who's not. So this is what we got, and then we started to analyze directional circuits, computation of direction, selectivity, and so on, and this is published. Now, the update I want to give here, and that is important to me in this context, is also to realize that this is not how the brain looks. This picture, I have to say, when I entered neuroscience, was plausible to me. I thought, okay, fine, yes, nerve cells, a little bit of interstellar space or whatever. Okay, the firing, I was aware that firing was not as literally meant as it sounded, but yeah, so there is activity, and but these cells, like this. But this is absolutely not what we have in our brains. The actual data looks like this. So this is the actual segmentation of a dense neuronal network, again, from the retina at this point, but we're starting to get similar things from cortex. Highly densely packed neuropil. All these cables are on the scale of 100 nanometers or so in diameter. We are flying through a tunnel, which is a blood vessel that we took out. Okay, so when you really dive into the neuropil, as we call it, the nerve tissue, this is what you get. And I think this is really important to realize when we are talking about building computational systems. Again, I'm not saying we have to do this, but what I'm trying to remind you of or put out here is this is how evolution solved it. Again, there could be better solutions. Again, we don't need to be birds to fly, but we should be, maybe appreciating the level of complexity that's used by nature to build systems that can compute in the efficient, energy efficient, and label efficient way that we talked about. Okay, just to give a little bit of an update, also a little bit of technological excitement. This is basic research, obviously, but this is happening, again, not somewhere in the world. This is happening in the middle of Europe. The fastest electron microscopes are those scanning with 60, 90, in the future, hopefully hundreds of beams, built by Zeiss, a German company. 3 of these microscopes are in the Max Planck Society in Germany. There are 5 worldwide, so we have 3 of 5. This is good. We get very large data sets, not just of mouse, also of human. I'll talk about that happily some next time. But this is happening. Data analysis is a big challenge. We use a little bit unconventional tools to combine, again, AI and human interaction to analyze our data. That's a separate story. But I want to come back a little bit to just give a small glimpse on what we mean by analyzing these networks. And these are early days. So this is just happening right now. So when you think about brains and so natural computers, if you want, and our man-made computers, then on the architecture side, it's very clear what the differences are. We talked about the cable stuff, but even more abstractly speaking, the key thing about our current computers is that memory and processing are separated, as you know. And this is absolutely not how the brain does it. The nervous system combines storage and processing. We have absolutely no evidence for a separation of the two. So that means it's really a different computational architecture. And that also means, by the way, that looking at the structure can be much more meaningful than looking at the structure of a computer chip. Obviously, if you slice up a computer chip, all you learn about is the microprocessor architecture. That's not going to tell you what program is running. That's not going to tell you what software algorithms are implemented and being used on this processor. So, but when we look at brains, looking at the networks means reading out potentially both the algorithms and the storage. And this is why we think this is so exciting. There are many more differences. Actually, brains are slow, as you know. If you want, the clock is more on the micro to millisecond scale. But they are highly recurrent, our networks, and operate in 3D. But for the last couple of minutes, I would like to focus on another aspect of brain architecture, and that is the existence of inhibitory nerve cells. So that's another difference, right? I mean, currently at least, standard AI is not particularly looking at any concept of inhibition, at least not explicitly. So you have, activity that can be high or low, but negative activity is not really something that is implemented. I mean, I'm not saying exclusively, but in most things that we know of. So, but brains have that. So we all have roughly 20%, 20 to 25% of our nerve cells, a fourth to a fifth, are inhibitory nerve cells. What do we mean by it? When they are active, they dampen the activity of the postsynaptic neuron. They, transmit GABA, as it's called. It's a particular transmitter, a chemical substance that leads to chloride influx, and so on and so on. The point is, when you activate an inhibitory nerve cell, one of these, 20-25% cells that we own, the next cells will decrease their activity. Now, obviously, that's kind of plausible that you need something like that to prevent your brain from going crazy, from becoming epileptic. Fine, that's definitely one part of the story. But maybe there's much more. And I think it's fair to say we don't know yet fully what this means. And I just want to show you a little bit how we start thinking about that. So this feature of the brain, which is called inhibition, means there are a population of cells, as I already said, that have a negative impact on the postsynaptic activity. And there are obviously 80% of the cells are doing what's shown here with this positive sign, so are exciting each other. That's what we know from artificial neural networks. That's the same story there. But these inhibitory nerve cells are special. And when we look at their activity, obviously one reason you could do that, in addition to preventing epilepsy, is that you simply overall reduce the energy load on your system, right? So if you are able to encode negative activity by activity of other nerve cells, then essentially you're reducing your overall activity by a certain factor, depending on how active these are. So this could contribute to energy efficiency. I don't think it's the key deal, but it is at least one aspect. But circuit-wise, what is it? So in a recent study where we analyzed the circuitry in medial entorhinal cortex, a part of the brain occupied with spatial navigation and so on. So what I'm trying to do here is run by medial entorhinal cortex, partly at least. And there, unrelated to the spatial activity, we think we found a very fundamental aspect of these circuits that made us wonder. And we found that synapses along the cables in our brains, in this case, a mouse brain, I should say, are extremely precisely placed along the cables, much more than we thought. I mean, we knew for some insects and other species that this could happen, but for a mammal, this was extremely exciting. And the key thing was that these synapses were used to always create what we call a triadic circuit motive. And this was much more preserved than we thought, in the sense that whenever an excitatory nerve cell contacted another excitatory nerve cell, always there was an inhibitory neuron in the loop. Not sometimes, but always. And always the exact same connection was happening. So this cell excited the inhibitory cell, which would make sure to inhibit the very same cell also contacted by the single presynaptic cell. So what we call cellular feedforward inhibition, because it operates at the level of each and every nerve cell. Extremely specific. And now we found that this is actually a closed gate. We found that the biophysical properties of synapse sorting, thick cables, fast cables, and a few other tricks make the circuit in a way that when you have a very active population, there is no transmission of signal to this excitatory population because the interneurons kick in. Now that made us wonder, why would you do that? I mean, obviously you can say, fine, I can build a gate easily, but this is very interesting because now you could think about how can you unblock such a gate, right? Do you have a switch signal there? Do you have expectations that come in? And this is really what we're thinking about, whether this is essentially a very basic hypothesis circuit where you encode by wiring all the things that could happen, but you're not activating it. You would go crazy if you were thinking about everything that could happen all the time. And only when information comes in, like I'm now heading in that direction, or more abstract context, like whatever, it's a not so sunny day or something like that, that makes me want to take my umbrella, that then this contextual information unlocks the most plausible hypothesis laid down in this highly parallel circuit. Now that's going very far. What I'm just talking about are hypotheses. I'm not talking about something that we've proven to be there, but what we've proven to be there are the circuits for it. And this is really a first step, very, very early, and first step, I think, in that journey to use circuits in the brain to understand how it's operating. And this could lead to novel computing, but again, this is a proposal I laid down a few years ago. We could maybe learn from these recurrent networks in the brain and maybe help build a more exciting technology also in terms of artificial intelligence. And again, we don't even know currently on what scale of recurrency our mammalian cortices really operate. This is something we're right now trying to find out in local circuit reconstructions from mouse cortex. Is it, is there anything like LCM? Probably not exactly, but on what level of recurrency are we operating? What are the specific circuits that we find in each and every animal, and obviously also in human brains? And with this, I want to conclude. We think that neuroscience inspired machine learning 70 years ago. That's clear. And then for a long time, the two fields a little bit separated, partly because computer science wasn't really able to compute with these ideas, and partly because we were occupied with other things. And then, last 2 decades or so, there was an inverse thing where machine learning became so powerful that it helped us a lot with data analysis. We're using a lot of machine learning for our image analysis. That's a nice twist here. But now we think is the time where we at least want to try by mapping these circuits, which we'll do anyway, but we'll ask questions about them, like the ones I've shown you, again, early steps, that may lead to ideas about how the brain is able to be so energy efficient and so label efficient. And this is what's happening right now. And again, it's happening in Europe, it's happening in Germany, it's happening here. So it's something that obviously could inspire totally new technologies, and I hope may contribute to some transformative developments in this field. This is my summary. Thank you very much for your attention.
We have a little more. We actually have a little bit of time left. Who would like to ask a question? Ines is first.
Thank you so much. Being a neuroscientist myself, I have a question about, well, I think usually we only incorporate the electrical signals, but do you see a chance of incorporating the chemical signals as well?
Well, I mean, as you know, the transmission from one neuron to another is always electrochemical, right? So you have the electrical part, which is fast and goes to the synapse, but then the 10 nanometers or 20 nanometers from the axonal terminal to the postsynaptic neuron are chemical. It's diffusion, as you know. So in that sense, every signal transmission is electrochemical, because then postsynaptically it's again electrical. So in that sense, it's all electrochemical, electrical. If you're talking about things like modulation, so state-dependent, as we think, transmission of substances that change the entire state of the brain, very simply put, that's obviously also something that is very relevant, for instance, for reinforcement learning. Again, another topic inspired by brain science, where dopamine is the key player. So that's obviously also a very highly researched area, reward encoding.
Okay, is there no other question? Oh, I'm not sure. He raised first. Give it to him, not throwing. Yeah. Safety first.
Hi, yeah, just the clock speed, 100, milliseconds for the brain, biology does it in milliseconds, and nanoseconds for silicon. Can we, if we sped the biological up to nanoseconds, would we lose something?
Oh, I'm not sure that's true. I'm not sure it's possible, at least with the current device. I mean, even at the level of proteins and the need to transmit an ion through a channel, which is at the core of our electrical activity, you have the 8 nanometer membrane across which you want to conduct a charged ion. So I don't think that even that speed would allow for nanoseconds. So I think it's more a practical thing, but what I'm definitely interested in is it's a solution which is not just about making it faster. It's a solution where kind of slow is okay, because maybe, people talk about then obviously the parallelity of the system, the sparse coding, right? It's also not highly active. Didn't mention it, but only 10% roughly of our cells are active at any second. So we have an 0.1 firing rate. Will it help? Again, I mean, could you do it? I don't think you could do it. If you did, would it help? I mean, okay, there's another interesting thing to think about, if I may. What sets actually the speed that we need to operate at? And I think gravity is key, right? If you think about, I mean, the object we care about fall from trees, usually, or maybe mountains, but essentially that determines a time scale on which we need to be able to react. And if you take that time scale, then milliseconds is fine. I mean, we react within the fastest is 100 milliseconds if cortex is involved. It's usually fine for most falling apples.
Okay, is there another question? Yes, you have to stand up and raise your voice because we are running out of battery.
Thank you for the speech. I have one question about the inhibitory mechanism
Yes.
that you explained to us. In how far does that relate to negative weights in a neural network, or even thinking about rectifiers?
Yeah, I mean, yeah. So, I mean, the first key difference is that in the brain, in our brains, not all of animals, but in mammals, there are specific neurons, specific nerve cells only doing inhibition. So that was as if you were separating those with a negative weight from those with positive weight. So that's already different from what you usually have when you just get a negative weight. Interestingly, some species don't do that. Flies don't do that, worms don't do that. So it seems to be something, it's called Dale's law. It seems to be an achievement or development of mammals and some other species. Again, I don't think we understand why you would do that. I think that's the fair answer.
Okay. Thank you very much. Is there another question? A last question from the audience? Then there, sorry.
What current, because it seems that you really focus on the hardware, not so much on the software challenges and what current hardware initiatives are you most excited about? Because there are these Neuro-inspired chips by Intel and all of these initiatives.
So first, I should say I would like to emphasize again that I try to point out that the software-hardware distinction is true only for our computers, not for our brains. So in that sense, when you say I'm focused on hardware, I want to hope you don't mean it dismissively in the sense who cares about hardware? Fine, I just try to make that point again. So yes, in the brain, hardware is what we call hard and software in computers, at least partly. I mean, there's a little bit more, but partly. Now, to your question about current neuromorphic, if you want, approaches. I think they're interesting and they're trying to represent what we currently know about the brain. What I try to show to you is that we are only now measuring these circuits, and all the current neuromorphic initiatives have been based on knowledge that was available, let's say, 20 years ago. Good learning rules, SCDP and so on, plausible learning rules, but network architecture totally not matching what we see. So I think that is an update needed, and it might be that we need to go to 3D implementations. You know, if you want to build a network of other 1,000, you may by cables, by non-wireless transmission, by wire-based transmission, you may need to go 3D. You know, some people are thinking about that, obviously.